We could get to Mars tomorrow from an engineering perspective. Our astronauts would arrive sick, and I don't know if they would come home. Let's take longevity interventions and see if they can prevent the damage accrued from radiation exposure.
Rapamycin still is the best we have, and nobody has done better yet, except Ora actually has a small molecule that's better than Rapamycin.
We're seeing up to 60% lifespan extension. We found something
They shortened lifespan by 90+%.
and it turned out to be our favorite dewormer and potential COVID treatment.
These were all FDA-approved drugs. You could find things that dramatically shorten lifespan. You could find things that have robust effects.
Candidly, and this is a hot take, frankly, I don't give a damn.
But at the end of the day, who it? If it's making people live better, longer, healthier lives, the field as a whole had narrowed prematurely. Nobody continued to do discovery science.
We're focused on the same 3, 4, 5 pathways, the same 2 molecules. Ora actually has the receipts. We can show you ground truth images of animals living longer right next to the controls.
The Million Molecule Challenge is an idea that we came up with. It would be the single most impactful project in the history of the field for $5 million. Hey everyone, welcome to the OptiSpan podcast. I am joined today by Dr. Ben Blue, the Chief Executive Officer of Ora Biomedical. And today we're going to talk about Ora, the sort of philosophy around why we even need a longevity drug discovery platform, some of the data that's come out of the Million Molecule Challenge to date, and then a new initiative that Ora is putting forward around repurposing, to some extent, longevity therapeutics for radiation resistance and why that makes sense in terms of both disease indications in humans and then potentially thinking futuristically, space travel. So lots happening at Ora. Thanks for stopping by the podcast studio today. I know it's a long way from your office.
Yeah, it was a rough journey going the 20 steps from where I usually work, but I love it to be on this side of the wall, honestly. Yeah. Thanks for having me.
So probably most of our viewers aren't aware that Ora Biomedical is located right next door to OptiSpan. Mm-hmm. So we get to see Ben and the team at Ora on a regular basis, which is usually a lot of fun.
It's great until we have people that we're giving tours that are really worried about worms, because we do a lot of worm research on the other side of the wall.
Yeah. So awesome. So maybe let's just start for the folks who aren't familiar with Ora Biomedical in talking about, how Ora got started, the thesis. I'll let you tell the story through your lens, and then I can tell it through my perspective. But you've been part of Ora from the very beginning, so maybe tell us a little bit about that story. And before we get too far into this, full disclosure, I am a co-founder of Ora Biomedical. Ora is a company that was spun out of my lab when I was at the University of Washington. I have equity in Ora, so everything and Ben also obviously has equity in Ora. So everything we talk about today, look at it through that lens.
I was going to mention that as well, but I'm really happy to be here. And yeah, thanks. So, I've had the opportunity to work with you for going on 8 years now. So
has it only been 8 years? Jeez.
Yeah, I know, right? Well, it's easy because I know this because I started in your lab the same year that I met my now wife. And so that anniversary is somewhat dovetailed with it. Yeah. So the whole premise of Ora is that we wanted to look at new molecules and new chemical space that no one has ever looked at. There's been an incredible amount of work done across the field of longevity biology, looking at a couple of core molecules, some of which are our dear favorites, some of which maybe turn out to be bunk, but I'll let that discussion go to other folks. And so, I joined your lab because I was really excited about the prospect of someday taking a longevity therapeutic into the clinical market. For me, I always got excited about this field because of the potential impacts to human health. I was lucky enough. I had a super centenarian in my family. My great-grandmother was the oldest person in Alaska. So from early age, I was just really excited about the concept of how and why we modulate aging. So I joined your lab because of the cutting-edge research that was going on around high-throughput screening. Right as I joined, one of our co-founders, the excellent biologist turned engineer turned computer scientist turned metal musician, Jason Pitt, had started the version 1 of the Wormbot. And that really appealed to me because I'd been working on automated platforms for a long time. And so Joining at that point, I got to be a foundational member of the design of it. And then I took over a lot of the analytical pipeline. So using AI to automate data analysis and prediction. And so this tool was based around the idea that in vivo biology is super, super relevant towards disease understanding. We've seen a lot of drugs that start in vitro with a cell culture target-based approach, then fail in clinic. And so in vivo, we could start looking at whole animals, and we start with the humblest of animal, our favorite model system, C. elegans. They only live about 3 weeks on average, so we can do the highest capacity animal experiments on new longevity therapeutics, both single and combinatorial use that anyone's ever done. And so in 23, we spun out of your lab. We moved into this site down in South Seattle, and we've been running ever since.
Yeah. And I think implicit in what you said is that because worms have a lifespan of about 3 weeks, you can do experiments in a short period of time. The Wormbot allows us to do those experiments at scale, and we'll talk a little bit more about what that means. And lifespan really is the phenotype that we look at first. It's not certainly health span from human health, human medicine is more important right now, but lifespan really tells us whether or not an intervention is affecting aging, or at least if you only look at one thing, that's the best proxy. If you can significantly increase lifespan of a population, almost by definition, you have to have modulated the biology of aging. And the Wormbot allows us to do that at scale in a time frame that's amenable to discovery. So that was great. Thank you. I'll kind of try to keep it brief, but from my perspective, give now my sort of history of how we started thinking about the Wormbot in my lab. So, this really stemmed from my feeling that the field as a whole had narrowed prematurely, gotten way too focused around the hallmarks of aging. That really became the dominant paradigm, circa 2009, 2010, when the hallmarks were first proposed. And their hallmarks are great. They've been very powerful as a communication tool for the field, but they also have had the consequence that everybody got focused on the hallmarks. Nobody continued to do discovery science looking outside the hallmarks. And I really felt like we needed a tool that would allow us to figure out what else is out there other than the hallmarks of aging. And in thinking through what you would need in order to do, massively high throughput, unbiased longevity screening, I felt like we needed some sort of platform, for lack of a better way of saying it. And if you were going to look at I was thinking 100,000 at that point. Now we're up to 1 million. But I was thinking if you want to do 100,000 lifespan experiments, you're not going to do that in mice. Certainly you're not going to do it in people. C. elegans seemed like the right sort of middle ground between yeast and mice. And I felt like we had an opportunity to build something that would allow us to do C. elegans lifespan experiments at scale. You mentioned Jason Pitt, was a postdoc in the lab at the time, had an engineering background and expertise. And Jason and I sort of thought we could build a robotic system that would allow us to do automated C. elegans lifespan experiments. Built the first prototype using a make block kit from Amazon. Looked pretty janky, but it worked.
My first bot that I built only set one fire in your lab.
We don't talk about that. But, through iteration, have gotten to a point where we created this system that we now call the Wormbot that is very robust. Coupled that with image acquisition and artificial intelligence to basically fully automate the lifespan experiments once they're set up, once the worms are on the plates. And now, can screen, I think Ora, probably, you tell me what the capacity is. There's only 6 or 8 Wormbots.
We've got 9 Wormbots here running.
Okay.
Each Wormbot can do about 100 molecules a week at full capacity when we're running real hot. So we can do about...
But that's replicates, right? So again, to give people a feel for what we're actually talking about, how many replicates of each molecule is that?
Yeah, that's 5 replicates per molecule.
Right.
So when we see a signal, we see a strong signal. Right. And
so again, this isn't the typical screen where you just test each thing, one molecule, one time at one dose. You have the capacity to actually do real statistics and have some confidence about false negative, false positive rates, things like that. So 100 molecules per device per week. And then the experiments last, let's just say, a month.
Yeah,
is that about right?
That's about what we run.
So 100 molecules across replicates per Wormbot per month. Is that about right?
Yep.
Okay, so that means you can do about 1,000 lifespan experiments, full statistics per month with 9 Wormbots.
Yep.
So if you were to scale that up tenfold, you could do 10,000 molecules per month. And that's easy, right? So again, the idea that you would be able to screen hundreds of thousands or millions of interventions, it's really just a scaling factor. And as far as I can tell, there's no limitation other than space and, well, resources to do that.
Yep.
Okay. So that's the foundation is the Wormbot. And we'll talk more about how Ora has branched beyond C. elegans. I think that's important because that's one of the criticisms people raise. Do you have a feel for the number of interventions, let's just say small molecules, that have been tested with 5 replicates in the whole history of aging research? In any organism.
Right. I mean, we're looking at maybe a couple thousand, realistically. You know, you have C. elegans as a primary research model where people have done some, but the data is hard to look at. It's sketchy and it's not well validated.
Yeah. So, I mean, you can go to Drug Age. That's probably the best single place. And I think there are less than 2,000 drugs even cataloged in Drug Age. And the I don't know about the vast majority, the majority of those don't even have 5 replicates, right? So I think it's safe to say Ora, even now, has by far the largest quality database of longevity interventions that has more data than the combined history of the entire field of aging biology ever, right?
And can I touch on that? Yeah. So one thing that I think is really important for what we do is when we make a claim or propose data or show a lifespan curve, because that's what I think a lot of your podcast listeners are going to be familiar with when we think about aging. Places like Drug Age, places like, academic papers, they'll show a lifespan curve and say, this is how we did it. Ora actually has the receipts. So we have never deleted any of our data. We have never deleted a single image. So when we say that something works, we can show you ground truth images of animals living longer right next to the controls that are 2 centimeters over from them with the same genetic population. Right. And so I think that's really important that we've got that established trust within the field. And, that's, I think, really important for the field. Yeah.
So again, I want to talk very briefly about the idea behind the million molecule challenge. And then I want to talk about, what Ora has found so far. So full transparency, Ora has not gotten very far on the million molecule challenge. So the million molecule challenge is an idea that we came up with probably 2023, I think is when we put the paper out.
That sounds about right.
Where we decided that it was a good target to set to quantitatively measure the effects of a million interventions on lifespan in any organism, C. elegans being the place to start. And that if we were to do that, it would basically change the face of the field as a whole, because we would identify likely hundreds of interventions that are more effective than the best-in-class interventions we have today. And it would enable artificial intelligence to actually be used intelligently. And again, I hate to say it, I don't think anybody in the field, at least for intervention discovery, is able to use AI intelligently because we don't have the foundational data set to inform intervention discovery by AI.
Yeah, exactly.
If you had a database of 1 million interventions, you would have that foundational data set that would enable the models to actually function well, right? So that was sort of the concept, and we pitched the idea, tried to get federal funding, NIH funding, unsuccessful. And again, don't need to harp on that other than it's just really hard to get NIH funding for discovery science. And we'll just leave it at that. Spun Ora out. And I think, we don't know exactly what the price tag would be for the million molecule challenge, but I think if we had it all in one lump sum, 5 million would be enough to get there.
Yeah, that was about what the math worked out to.
And so I'm just going to, and so far we haven't raised the money, and that's okay. Like, I get it. People can put their money where they want to. I firmly believe that is a tiny, tiny price tag for the impact that this would have on the field. And frankly, I don't care if it's done. I know you're the CEO of the company. I don't care if it's done on the for-profit side. If somebody wanted to put up $5 million, we would do the work at Ora, make the database freely available, to the community and revolutionize our understanding of aging biology.
Yeah.
It would be the single most impactful project in the history of the field, I am confident to say, for $5 million. Okay, I'll stop there. But if anybody wants to help, you know where you can find me.
Can I highlight some successes from it that I think are really cool? So you're right. Like we've had about just shy of 150 individuals that have contributed.
Right. So maybe worth saying how that's happened.
Yeah. So we opened the Million Molecule Challenge up on our website. We let people just come in and sponsor an experiment, and we return data to them. We also made the data publicly available. And this was done somewhat piecemeal, where someone could just pick any intervention from one of the libraries we threw up on there. I think the library itself had about 2,000 molecules that people could choose. People often chose stuff that was like they chose rapamycin multiple times. We actually stopped putting it up on the website because we were like, guys, let's do something else. Let's do something else. Let's do something new. And so, I want to highlight one of the people that was really, really impactful was this fellow, Krister Kauppi, who runs Rapamycin Longevity Labs. And so he started funding all of these random mTOR inhibitors. And we actually found omeprazole is a novel, understudied mTOR inhibitor that we found had one of the strongest effects on lifespan that I've seen, to the point where now it's taken over as our positive control across all of our screening process. So when we do an experiment, just for context, we always make sure that we have a negative control, i.e. animals given the equivalent of a placebo, right? So just the solvent that the molecule's dissolved in. They're given the drug of choice. That gives us the effect size relative to baseline. And then we always give a positive control. That shows that the experiment actually has good scientific rigor and context, because the positive control should always work. And if you have something that works better than that, fantastic. And so now this novel molecule that came out of the Million Molecule Challenge is I kind of wonder if we're one of the biggest buyers of it on the market right now, because we are consistently buying it. And I want to highlight the work Krister's done, because he's then taken that and he's submitted it to the ITP and CITP for further research. And so
let's not talk. Okay, you brought up the CITP. I'm really sorry you did that, but I have to make this point. The CITP has cost $20 million since existence. We could test 1 million interventions for a quarter of that. This is
I know we have different feelings, and for full disclosure, one of my earliest projects was working on the CITP building.
We don't have different feelings. Do you think that was which would you rather have? 4 million interventions tested, or what the CITP has not accomplished to date?
In a heartbeat, I would say I would rather have 4 million interventions for it. I think I have less strong feelings around some of it, but I will say, I think that getting the validation that Krister's done by moving it into these federally funded programs is a highlight.
If it gets tested in the mouse interventions testing program, great. Absolutely. Okay, so again, $5 million, biggest impact of any single project on the field of aging biology to date.
And I think that's
somebody needs to do this. Okay, let's just move on.
No, I think it's worth highlighting that importance of having this in the space, because right now the aging field is trying to make the case that we as gerontologists should take drugs to market for aging and longevity, but we're focused on the same 3, 4, 5 pathways, the same 2 molecules that keep getting derivatized. And there is whole aspects of chemical space out there that are untouched.
Exactly. And we don't know combinatorial space, which the Million Molecule Challenge can accommodate.
Yeah.
I think it is worth saying, and I've said it before, so probably everybody listening who watches the podcast regularly has heard this ad nauseum, but rapamycin still is the best small molecule longevity intervention we have in terms of both magnitude of effect and replicability. And, it's been almost 20 years since we first published the first lifespan experiment with rapamycin, and nobody has done better yet, because frankly, nobody's really looking in any innovative way, except Ora actually has a small molecule that's better than rapamycin. So I know you probably can't tell us a whole lot about that molecule, but maybe you can just give us a little bit of insight into, what you've seen. And actually, we haven't said this yet. What is your ballpark estimate of the total number of interventions in the Ora database right now? Because it's relatively small. I want people to get a feel for how how powerful this technology is, but also how early we are in the game.
Yeah, the whole Ora dataset comprises less than 10,000 interventions, and so that's both single interventions, combinatorial interventions, and dosage curves for things that are interesting. So, we're still early into this space, but we've proven the principle that you can look in this unbiased way and find things that are really exciting. And the fact that we're finding things after having just looked at 10,000 molecules is so compelling because we're actually
not only finding things, but things that are better than anything that anybody's ever found before.
We're seeing stuff that extends lifespan in C. elegans at lower doses than rapamycin, up to 60% lifespan extension, which is
for single drug.
For single drug. And that's not even looking at dose optimization or chemical derivatization of it. So I think in terms of proving the case that high throughput unbiased screening is effective, we've made it, even with the modest amount.
Not that you should have to prove it, because anybody who knows anything about biology would expect that. But not everybody knows very much about biology, so I understand that.
And it's okay. The field is still figuring out. We're at the stage equivalent of Ponce de Leon. We are out there combing through the wilderness looking for the things that may or may not work. And I think that's
maybe not the best reference.
I know, because it didn't work out, but I love the idea of, exploration.
Yeah.
And I think that's the point, is that, we're figuring it out step by step.
Okay.
And
go ahead.
You made a great point talking about the limitations of AI. Right now there's 100,000 companies saying they're using large language models for intervention discovery. They're using open source trained data. None of them have the compelling ground truth data we have to train on. And so that's why we're excited to work with new and existing partners. Like, we just partnered with Rhizome Biosciences. They're an AI-based discovery. We're giving them, some blinded data, and they're telling us novel ways to look at it and novel ways to use the best-in-class AI to actually find the next derivatives out of it. So it's pretty exciting.
Yeah. Yeah. And I mean, again, I think that the larger the data set you've got to train the model, the more powerful these AI tools are going to be, the more accurate they're going to be at predicting new interventions, combinations of drugs that have larger effect sizes. All right. So the platform is there. It's ready to go. Scalability is not a challenge other than resources to allow the scaling. Again, my feeling is, if Ora had $5 million put towards the million molecule challenge tomorrow, is probably a 3-year time frame to get the entire million molecule database completed. And I only say that because, we're in almost 2026. When we started this in 2023, if we'd been able to do it then, we'd be there now. Yep. So that's not a long time for that sort of level of payoff, with the recognition that there are going to be discoveries along the way. So again, maybe not. That'll happen in 2026. I'm going to be cautiously optimistic we'll really be able to pull the trigger and go. I know you've been thinking about other sorts of approaches, and I want to get to that. Let's first talk a little bit more about what you're able to do with the data that you've already collected. So you've got some hits that by themselves have large effects on lifespan. What have you done in terms of testing combinations of molecules? And again, I know you want to be careful not to give anything away. I know some of this is in collaboration with other partners, but just in a general sense, like, what do you see when you put combinations of things together?
Yeah, it's really interesting. I think one of the cool ways that I like to think about combinatorial interventions is, we don't know yet the full complexity of the biology that happens when you intervene in an aging system. And so, because there's so many different nodes that are important to touch on, we've found drugs that by themselves have no effect. You combine them with one other molecule, and suddenly you see an incredible effect.
Either direction, right?
Either direction, right?
Yeah.
I remember when we started this in your lab, the first week of screening, we found something that killed the worms right away. And this was, and for context, this was in 2020, it was before we spun out. It was in 2021 or 2022.
No, it had to be 2021, because I remember the context here.
So this was still during COVID. There was a lot of noise in the scientific community about how to deal with COVID still lingering.
And we were testing an FDA library, or at least part of an FDA library, right? So these were all FDA approved drugs. There was this thing that shortened lifespan by 90+%.
Yep.
Right?
They were basically dead 2 minutes after they hit the auger. And we always do our experiments blinded. So we don't know until we go and decouple, the coding of the experiment, what everything is so that we don't get biased. So we decoupled it and it turned out to be
Ivermectin.
Our favorite dewormer in potential COVID treatment. And I can tell you as a scientist working in the field, I was thrilled to not have to publish that Ivermectin extended lifespan.
Or shortened lifespan by 90%, as the case may be. And as it turns out, I mean, we're kind of making fun of because of the whole COVID Ivermectin thing, but it makes perfect sense. And in some ways, it was actually very reassuring to see Ivermectin come out because it is a known anti-helminthic. It kills worms.
Yeah.
So
it dewormed our experiment.
So it actually makes perfect sense. And I think points to something I hadn't really appreciated until that point, which is that when you go testing 1 million interventions for things that increase lifespan, you're going to find other stuff that's interesting and valuable, like novel anti-helminthics. Maybe, and again, we haven't really pursued this, but you could easily imagine a combination of natural products, for example, that have a very potent anti-helminthic effect. They could be very, very useful in livestock agriculture, right? Anywhere where worms are a problem, you could have value in something new that you find. And it just comes out of the process of looking for, because this is being done quantitatively, right? Where we're getting full lifespan on every intervention that's tested, you could find things that dramatically shorten lifespan. You could find things that have robust effects on activity or chemosensation. Those will come out of the data set that's being collected, which is potentially going to inform basic biology and have sort of interesting market potential.
Yeah. And that's the way that I think about it. Anti-helminthics is a great touchstone point because, think about how many millions of dollars are spent on that each year in the US alone, agricultural wise. Yeah. Think about the cost of human suffering in the developing world where anti-helminthics are drastically needed and not necessarily cost effective to get to everyone that needs it.
Right.
We have the potential to find things that can find access to those markets and access to those patients to do real good effectively.
Yeah, and there are, C. elegans is not parasitic in any way, but there are related organisms that are parasitic. You could imagine, again, we don't have this because we haven't had the scale to look for it yet, but you could imagine a combination of natural products or a pharmaceutical that could be taken orally that would be very potent at killing parasitic worms that are causing problems in people and animals. So again, there's lots of additional value that comes from doing this sort of a project. To go back to the combinatorials, though, the context of that screen, if I remember correctly, wasn't just to look at FDA-approved drugs by themselves. It was to show that you would get, or to maybe explore, because we didn't know at that point, how often you got unexpected interactions. So it was everything got metformin, and then everything got metformin plus one other FDA-approved drug.
Yes.
And my recollection is we got all sorts of weird, I don't know if weird, unexpected synergistic, anti-synergistic interactions, which kind of proves the point that you're going to get unexpected things when you combine just only two, in this case, FDA-approved drugs.
And so I think that's such a cool point because, one of our other co-founders, Jan Gruber, has really led the field in the aging space, looking at predicting what two compounds based on their molecular signatures should work together to produce interesting effects on aging.
Right.
But the reality is no one in the field has touched on this.
Not at scale. Jan's done, maybe 10 or 12 experiments published. I'm sure he's done more in his lab,
but like the ground truth from that has yet to be explored. And you can only do that when you have the scale to do 100 compounds by 100 compounds.
Right. And I think one other thing that's worth mentioning here is, like the small scale experiments Jan has done is combining things that already are known to extend lifespan, right? And just asking, where do you see additivity? Where do you see anti-synergy, whatever. Yep. But a lot of people are taking multiple supplements, multiple medications, each of which they hope will have a beneficial effect on health span or lifespan. But we really have no idea what the interactions are going to be. And what the metformin experiment that we did really solidified in my mind is it's really hard to predict where you're going to get negative interactions, other than it happens maybe more often than you would expect. And I've heard Brian Kennedy, also one of the co-founders, say that when they do this in mice, they more often see counter effects. So in other words, where each drug by themselves might have a positive impact on health span, but when they combine them, they offset each other, so you get nothing. They see that more often than they see any sort of additive effect. So I think this just points out again the need for a very large data set of interactions so that we can then hopefully use AI intelligently to predict,
right,
with some level of accuracy, interactions of novel combinations.
Exactly. And I think that, one of the things that I think is so fun and exciting, and I talk about discovery and exploration because that's the way I like to think about what we do. We've partnered with groups that are excited about natural products. And so we've screened a bunch of those, and we've seen incredible successes based on those screens and also incredible failures with things that are publicly available. And it's shocking to me that right now, there isn't as good data out there showing, basically what we've seen when we've worked with our partners, where their natural product is as effective as some of the best-in-class aging therapeutics that are out there and available today.
Right.
That just shows the way that the space is unexplored.
Yeah, and there's so much to learn. And again, I think most people, even people in the field who should know better, don't really appreciate how little is actually out there. And of that, how little is actually high quality data, meaning it's been replicated multiple times, done under standardized conditions. So there's a tiny fraction of what I call the intervention space that's been explored, and this huge opportunity to really see what's out there.
I had a really interesting conversation a couple of weeks ago when I was talking to someone, and I showed them a little bit of data about one of, not our most exciting molecule, but one of the ones that we see some novel applications for. And they asked me, well, you're doing your screening. What's the mechanism? Candidly, and this is a hot take. Frankly, I don't give a damn.
Yeah, I'm with you.
That's kind of the premise here, is that mechanistic stuff, that is easy science conceptually. You look at the pathways, you look at, you do some knockdowns, you do some gene expression data. But what's more exciting is seeing a molecule that has a real 40% lifespan extension effect on a video of animals in front of you.
Yeah, and again, I think so being a little facetious, I think both of us do care. Like, again, you want to understand the biology, you want to understand the mechanism. I think unfortunately, this is a symptom of what I was referring to earlier, where the field became very narrow very quickly, and everything is about mechanism and nothing is about what actually works and how well does it work, right? And so there needs to be that balance. And again, when you want to actually have an impact on human health or companion animal health, to get through the regulatory process, you probably need to have some understanding of mechanism.
Absolutely.
But at the end of the day, who gives a shit if it's making people live better, longer, healthier lives? Who cares how it's working? As long as the benefits outweigh the side effects, that's what really matters. So
absolutely.
So all of this focus on what's the mechanism, what's the biochemistry, I think it's counterproductive when it gets to the point where it becomes the only thing that matters. And that's really the only thing that matters when it comes to getting grants at NIH these days.
Yeah. What kills me is when I see, and again, we start in worms because they're easy and fast. Like, there's so much work between a worm and a human, but people use them to do mechanistic studies. And what absolutely drives me insane is when you have an otherwise cool paper about intervention, they say, we saw a lifespan extension. They show a 10% statistically significant because they looked at,
probably
200
not
animals.
Replicable if history is any guide.
But, who gives, the premise here is that C. elegans is a great starting spot, and it's a great way to find signal from noise. Yeah. I'm not convinced that a 10%, 15% lifespan extension is worth even doing the next steps of preclinical discovery.
I agree.
I care about seeing stuff that has 40, 50, 60% lifespan extension.
100%, 200%, yes.
Because if it's working in a worm at that capacity, the biological effect is happening in vivo. Let's then take that into cell culture. Let's validate
or go to mice. Again, this is where maybe I think you and I differ a little bit, because you're thinking about this more from the perspective of what are investors going to invest in. I'm thinking about it more from the perspective of what actually matters, right? And so I think if it has 100% effect on lifespan in C. elegans, and then you go do the mouse experiment, take you 6 months. If you start with old mice, you're going to know the answer, at least for health span. If it's having a big effect on health span and it has a 30% effect on lifespan in mice, you've got something as good or better than rapamycin. Go.
Yes,
right?
absolutely.
And both are valid viewpoints, and I think definitely in parallel, showing that you have biological effects that are interesting in human cells has a lot of value and gives you additional confidence about moving on from mice at that point.
Yeah, and I think it's I like the difference of matter of perspective because, I think you're coming at it from the perspective of someone in the aging space in general.
I just think investors are myopic and aren't really paying attention to the big picture. And I understand that's the reality.
And and I think it's true. I feel like, I am on the very translating discovery into the clinic side right now, where I'm talking to investors every day, and I'm making the case that we can take something that has groundbreaking longevity potential, but I need to have something that I can show them that says, but it's going to be into clinic in 2 years for a billable indication that we can actually get into space. And eventually, once it's in the space and we get this beachhead market, then we can start doing the exciting longevity stuff. But unfortunately, I have to take a very grounded approach.
I don't think it's unfortunate. I think it's the reality of how you move forward in the biotech space.
It's true.
It's good. Okay, so let's talk briefly about the Million Molecule Challenge.
Yes.
So there's been some data generated. That's still running, right? If people want to participate, they can nominate interventions through the website. They will go into an open access database. So that's one of the unique features of the Million Molecule Challenge, right? Is that anybody can get access to the database. Do you want to talk about that and how people participate and how to see the data and play with it?
It's right now it's on our website. We've got the interventions.
What's the website?
orabiomedical.com/Million Molecule Challenge leaderboard. But
I'm sure if you just go to orabiomedical.com, there will be a button somewhere on the homepage to find the Million Molecule Challenge. You can see it,
and it's got a couple hundred single interventions as well as combinatorial interventions that people have studied with all the data on there. One of the things I like, I love science education, so I've actually given the raw data to a couple of high school groups where students can then look at it and use it for discovery and potential. We had a kid that worked with us get ribbon at the state science fair last year.
What state?
Washington.
Okay.
Thank you.
Asking. Yeah. This is an international project, right?
I know. And we've had people from all over the world come in and work with us. I mentioned, and I will keep mentioning Krister. He's an incredible advocate for longevity science. And he took our baseline idea here and was like, well, I want to just look at mTOR, which is very valid because mTOR is one of our favorite pathways. But if you want to do the focus screening, sure. And he actually got us to fundraise up to 600 novel mTOR inhibitors. And we're making that data publicly available as well. We've already published 300 results from that, and we're going to be publishing 300 more as we continue to crowdsource that campaign.
Yep. And again, I'm not going to ask you to give away any of the names, but I think it's fair to say there's more than one mTOR inhibitor out there that works much better than rapamycin for extending lifespan, which again, anybody who understands biology would say that's obvious, but that probably will surprise a lot of people. Ora has found multiple mTOR inhibitors that are better than rapamycin for longevity. Yep, that's important.
And I think this speaks to the kind of the scale at which we work, where, right now, every single person that's done it has been a meaningful contributor to longevity science. But, to mention the scale that we've talked about and wanting to get to that place, we are looking for the visionary that wants to come in and fund the best possible
library.
Change the
game, guys. All right.
Yep, I know.
So the Million Molecule Challenge database is out there. People can get access to it. And then people can still participate.
And you get a personal report from one of our scientists showing you your lifespan curve,
right,
even before it goes online.
Right. Now, do people have to pick a molecule, or can they just say, do whatever you think is the best science?
Yeah, they can just be like, I want to contribute. That's great. Let's go for it. We love that. And we still put the molecules at random up on the leaderboard that they've sponsored.
So people can participate that way. People could, one or more people could fund the entire Million Molecule Challenge, or Ora is raising for investment. Full disclosure, shareholders, yes, we are trying to get people to invest in Ora because I think this is important. So people who are interested could also come in as an investors. If the Million Molecule Challenge was funded through an investor, it probably would not be made publicly available. That's just the reality, right? So again, I would love to be able to make an open access database with 1 million interventions available to the community, because I think that's how the biggest impact happens.
Yeah.
This is the real world. We're adults. If it's done in the for-profit sector, it's done in the for-profit sector. Whatever. It needs to be done.
Yeah.
Okay.
Hear me out. I just had this crazy idea. Okay. Brian Johnson, if you see this, we'll test every single metabolite that you record in your body in a personalized C. elegans strain.
And I will stop making fun of Brian Johnson.
So
Maybe.
I am impartial.
Actually, you know what? We are not going to go down the road of asking Brian Johnson for money.
And I choose to be friends. I'm friends with everyone. But I think that's the point, and we are, this is where we dovetail both sides of what Ora does. So we have our platform. That's the million molecule challenge. That's generating this database, and it's the high throughput discovery. On the flip side, we have the targeted approach. And oftentimes when I'm talking to investors, like I said, I'm saying, we're going after this specific indication that's going to see a return that then we're going to expand from.
Right. So again, one of the beauties and the challenges about finding interventions that target biological aging is you have dozens of endpoints that you could go after from a clinical trial perspective.
Exactly.
So it gives you a lot of opportunity. It also means you have to be somewhat strategic and thoughtful, and also, I think from an investor perspective, hone in on one or a few to really tell a compelling story about how this intervention is going to move through the drug development pipeline, the clinical trial pipeline, eventually to FDA approval, assuming it's a novel pharmaceutical. So I know you and the team at Ora have, thought about a variety of different ways you could go after this. And I think one of the newer areas that has become a focus, and I think for good reason, you've got some interesting preliminary data is around radiation resistance.
Yeah.
Right. So why don't you take us through that and how you landed there and why you think that makes sense.
Landed implies we're returning to Earth. I like to think that we're taking off from there. Sorry, bad joke, Matt. Matt usually gives me a pretty hard time about puns, so I'm trying to rein it in today as someone that's worked with him for so long, just so that, he still publishes this podcast. So, full candor, I started out as a giant space nerd. When I was 5 years old.
That's shocking.
Yeah, huge, right? I feel like I've talked to him on multiple podcasts of why I think Dune matters to the longevity community so much. I won't do that today, but I've always been really fascinated with space and space exploration, where we go as a, species. So, last year we were seeing data results roll in, and I had this, I think, admittedly slightly off the wall idea that we should look at space health and space aging. So astronauts and high altitude pilots experience much more radiation, and they've actually shown signs of accelerated aging. So what if we flip the script? And this is the idea that I proposed to the US Air Force and Space Force. Let's take longevity interventions that we found through their effect on lifespan and see if they can prevent the damage accrued from radiation exposure.
Yeah. Just to be clear, I think radiation certainly is part of the accelerated aging for space traveler and astronauts. Lack of gravity is probably also contributing, right? Yeah. So it's not only radiation, but radiation certainly makes sense.
We picked one aspect.
Yeah. But I also think it's worth, maybe mentioning that there is a long history in aging research of longevity promoting interventions also inducing stress resistance to a variety of types of stress. So ionizing radiation, thermal stress, oxidative stress. And so it is somewhat intuitive and certainly backed up by a breadth of data that looking at things that extend lifespan is a good place to start if you're interested in stress resistance. And so it makes sense that you would enrich for things that confer resistance to ionizing radiation.
Exactly. And so we were really honestly lucky, but I think we found advocates that really believed in longevity medicine. And so we partnered with the US Air Force because this is so impactful for astronaut and pilot health. Right. And so we ran a screen where we irradiated the worms, starting with UV radiation, full disclosure, not the high energy cosmic radiation that's also Ori, but DNA damage is DNA damage fundamentally. There's different types of it. There's different mechanisms.
Right.
We care just initially about this first as our first.
And UV radiation obviously is important for people on Earth, right?
It's the lead, it's the lead driver of skin cancer, right? And so, there's a whole aspect of avenues where I want to take this research and where I'm going. We're focusing acutely on radiation resilience in the context of people that are undergoing radiotherapy. And so right now we've found some really exciting molecules that improve resistance to radiation by acting on aging biology and aging pathways. So from my talking to investors perspective, that starts with this really acute treatable market. But where I envision this going and where I think the science is backing it up is that we're starting to see therapies that are not just impactful for this acute disease, but are letting us have longer duration stays in the International Space Station. They're getting us to Mars eventually because...
Elon, are you listening?
The reality is we could get to Mars tomorrow from an engineering perspective. Our astronauts would arrive sick, and I don't know if they would come home.
Yeah, right.
That's the premise. And so longevity medicine has this incredible potential to be a game changer, not just on the longevity side, but on the human resilience side. And so, we found this kind of niche market here that we're going after in terms of radiation resilience. That's not the only one.
Yeah. So take us through, so you get a hit in worms, it extends lifespan, it confers better survival after exposure to radiation in worms. Yep. What do you do next to convince yourself that there's a there?
Yeah, I know. And the there is the important part. So right now we're looking at, we take our in vivo hit, we move it into in vitro cell culture.
So these are human cells.
Human cells growing, and then we look at DNA damage markers through a variety of cell culture techniques. So looking at the accumulation of DNA damage repair mechanisms, which mechanisms are going to the cell, how long do those mechanisms stay active, what's the efficacy of them. And so, following this, what I'm trying to do in the new year is actually take this and then go back in vivo, but go more complex. So take this to a mouse model and look at how well does it promote radiation resilience in a UV sensitized mouse model. Right. That, I think, is going to be the linchpin for some of this story and for some of the science showing efficacy. Right. And so, the reality is we start with the humble worm. That's not enough, right? We always end up taking it to a mouse model or to another more complex mammal system before we ever think about people. And so, that's some of the next steps. And, you can see the trajectory of Ora is a trajectory of many biotechs, right? You start with a signal and then you go in and you advance it into more complex models.
I think the difference, and you've alluded to this, but it's probably worth double clicking, is that in Ora, the signal comes in vivo, in an actual animal, right? And so again, whether in vivo in a worm is going to be more likely to translate through to in vivo in a human compared to in vitro cells in a human translating through to in vivo in a human, we can't know because nobody's ever really done that direct comparison. My intuition is when it comes to longevity, because that's driven by the biology of aging, in vivo in a worm is actually going to translate better through to in vivo in humans than experiments done in cell culture looking at the hallmarks of aging.
Yes.
It's an intuition. I don't know if it's true, but that would be my guess. And if somebody were taking odds on that, I would put money down that will be the case.
Yeah. And I mean, it speaks to, I think, some of the hubris in the field that we know how people age and we know the molecular mechanisms.
Right.
The complexity of human biology is so poorly understood at this stage, despite our best efforts. And so there are novel interactions and novel combinatorial interactions that we can't predict with AI yet because we don't have that data.
And you can't model them in cells in culture. Or I mean, they just don't happen in cells in culture, but they do happen in a whole animal context. Yeah.
And you look at the failure rate of in vitro studies where you find a molecular target and the molecule works incredibly well in a human cell at one metric that you're measuring, maybe two. But then when you put it in an animal, it's toxic at that dose. Or it
doesn't get delivered to certain cells and certain tissues, gets metabolized by the liver, so it never reaches the target tissues. There's all these things that can happen, right? And again, nobody's saying worms are perfect.
No.
And this isn't, I mean, I think this is an interesting aside. There is this bias, I think, again, from people who don't really understand biology very well, that most things that extend lifespan in worms don't work in mice. There's like no evidence to support that. Yeah. If anything, it's the other way around. Like nobody has really done a really careful analysis of the things that are reported to extend lifespan in worms. How many of them actually are real?
Yeah.
And of those that do, how many have a positive effect on lifespan in mice, accounting for dose response? Like those experiments just haven't been done. So there is really no way to know the answer to that question. So the people who go around talking as if most things that work in worms don't work in mice are just talking out of their asses because we don't have the data. I can't say that they do, but we definitely know about a fair number that work in worms and also work in mice. And so what other things are you thinking about in terms of resilience, right? So again, clearly longevity interventions are going to enrich for things that promote resilience. There are a lot of directions you can take that. Is there anything else that you've been working on at Ora along those lines?
Yeah. So one of the things that I'm really excited to build on, we've mentioned working with the U.S. Air Force and U.S. Space Force. So what we're seeing is that there is real use case scenarios for patient populations that exhibit chronic stress, chronic environmental insult, and that's people working in the military field, either warfighters actually in field or even people that are undergoing training. With a special focus on some of the special operations forces because they're the oldest warfighters and they're basically professional athletes. But they've spent so much time training and investing in these people that a longevity therapy that can potentially give them a couple more years of service, make them recover faster from injuries that would otherwise take them out of field is actually really, really promising. And so one of the directions that we were talking about with the folks working on chemical, biological, radiological, nuclear defense is, well, if these people are exposed to environmental toxins, just like we think of radiation as a toxin or starvation potentially as an environmental harm, heat shock, could we model the effects of something that maybe is more coming out of the space of chemical warfare? And so we actually worked with them to select a couple of novel targets. So in this case, the mildest form of common neurotoxin. And we tested in the animals and we saw that we could actually modulate resilience with therapeutics.
So again, just to take through this, you treat the animals with the neurotoxin and then ask whether or not an intervention can attenuate the negative effects of the neurotoxin.
Exactly. So can you, we know that we resist environmental insults and toxins. Toxins gets into kind of that phrase gets into.
There's a variety of stresses you can use. This is really just another form of stress. So, you got radiation, oxidative stress,
organophosphate neurotoxins. So, and so they're also widely used in the farm industry as well.
That's what I was going to say. I mean, I'm guessing that there's a variety of people who are exposed to these or similar types of environmental exposures where this could be very relevant. So, farm workers, you mentioned, I would imagine first responders, probably. I'm sure there are a variety of others. And I think this is interesting because we've never really talked about this before, but, it opens up the opportunity, if you've got, let's just say 100 longevity interventions that are very effective, you could take that 100 or 1,000. If you did 1,000,000, you'd probably have 1,000. And you could then screen those as a subset across a variety of different environmental exposures, right? And find which longevity interventions are most able to increase that particular stress response pathway to give you the largest effect for that particular environmental exposure or toxin or whatever stress you're interested in. I mean, there's a huge opportunity here to spin out a variety of programs looking at specific indications for specific types of stress. And it's really a resilience program, but it's personalized resilience because you're looking for specific stresses.
And it kind of was inspired by a conversation we had years ago where when we were kind of thinking about the million molecule challenge, the other option was to pick a suite of disease models. So, we actually have a lot of data with a model of proteotoxicity kind of founded on the idea of Alzheimer's disease.
It's an amyloid beta model.
Yeah, your listeners know about it. And so we did some original screening looking at modulators for that. We also have looked at models of Parkinson's and some other age-associated diseases. My hypothesis was to take this in a slightly different direction and look at the suite of things. So we've actually tested neurotoxins, we've tested radiation, we've tested a whole host of other associated stressors that are relevant to kind of our daily environment or acute injuries that happen in the course of certain workplaces. So this is something that we're really excited to work with folks on and continue to develop.
I'm interested, and again, this may be a place where C. elegans isn't a great model, but I know people do model compression in. So traumatic brain injury, but in a worm, you're doing it in whole body, right? So you use some sort of compression model. And I'm guessing longevity therapeutics would confer resistance.
Exactly.
Some of them to that.
And this is a way that I think about this in the context of describing what we do, not necessarily the specific molecular pathway, but, we think as we age, it's harder for us to recover from injury. So if we can attenuate aging, we'll recover better.
Yeah, because you guys have done some wound healing stuff as well as following up on a few of the hits in a cell culture wound healing assay,
exactly.
Right?
And so that's one of the places we're thinking about it. I'm getting to work with a fellow trainee of mine from our time at the University of Washington on the Alzheimer's disease program that has a mouse site in Texas. And so he's worked with the DOD or DOW before, and we're actually going to be looking at taking some of our injury resilience or resilience to injury promoting compounds and looking at them in mouse models.
Yeah.
Everything into the mouse model.
Yeah. What I think is interesting, you correct me if I'm wrong, but the little bit of data that I've seen on the wound healing assays is, this is maybe a good example to where the longevity interventions are going to fall into a couple of categories, those that promote wound healing and those that might even impair wound healing, at least in this model, right? And mTOR inhibitors, again, at least at certain doses, do not seem to promote wound healing, right? Yeah. But other longevity interventions do. And that actually fits with, what we know that high-dose rapamycin can impair wound healing in animals and people. But I think that's a good example where not every longevity intervention is going to confer resistance to every stressor, but there will be subgroups based on their mechanism of action.
And I think we talk about where we want the field to be in the near future. And you can think about this as a way to start getting at the personalized medicine, where the longevity intervention suite of supplements and medications that you take for what you're experiencing may be completely different than what I need. Sure. And I think this starts to get at untangling that.
Yeah, I mean, it starts to. It's a complicated.
I know.
It's a complicated issue, but I do think if you had a large enough data set, you could absolutely then use artificial intelligence to model additional interventions and be able to predict which stressors are they going to promote resistance to, which might they actually be counterproductive. So it is a beginning to get down that path. And then I think combining that with predicting how these things are going to interact when you're putting them together starts to get you towards at least, something around individual response to different protocols, for lack of a better word.
Yeah. And fundamentally, it's a numbers game, right? Like that's really what we're proposing. So we can model all of these different types of wound healing, all of these different types of cellular resilience. And what we see is not every one of our best identified interventions works across all of these.
And that's exactly what
that's what we want. We want different things for the right solution.
Yeah. So one thing we didn't talk about, but I want to go back to with the million molecule challenge, because again, I think this just points to some of the emergent properties that come out of having very large data sets. Even though, in my view, the Ora data set is not anywhere near as large as it needs to be, it's still the largest that's out there, at least of quality longevity data by far, maybe by an order of magnitude. So one of the things that was done early and that I know you've updated more recently is to start to look across all of the interventions, their effect on longevity, on lifespan, and then you start to put them into pathways, right? Because for most of the things Ora's tested are either FDA approved drugs where we know something about the biochemical target, or natural products where at least there are ideas around what the biochemical targets are. So what does that look like when you start to then ask, if we group those by pathways, what is the effect of molecules that target pathways on longevity?
Yeah, it's really interesting. And one of the things we can show your viewers is that when we group things by pathway, what we see is a giant spread. And it's based around not just, we have to account for dose and for other,
right. And this analysis is fairly crude, right? Where it's just, you're not even worried about normalizing for dose. It's just each dose is its own unique intervention in that particular analysis.
Exactly. And so what we find is, if you look at, say, mTOR and PI3K and that pathway, it's actually one of our best on average
in terms of effect on lifespan, positive
effects on lifespan. But even within that, you see things that can extend it by up to 50%, which is incredible and speaks to the power of the pathway, but also decrease it by 30%. And that signals some of the issues with looking at a target-based approach to discovery, where you start with a pathway, and if you just look within that pathway, you're not going to necessarily enrich for hits. You're going to find things that don't work as much as...
Of course, especially if you can't do a dose response.
And so what we've seen now is that it kind of validates our original idea that we need to do discovery at scale to find the things within each pathway that are biologically relevant, because there's so many different effects.
And then if I remember correctly, and this sort of surprised me when I first saw it, that autophagy was the category that came out as shortening lifespan the most, which I guess makes intuitive sense if these are autophagy inhibitors, but most people think about autophagy as being beneficial for lifespan. But it makes sense, right? If you're blocking autophagy, you should shorten lifespan. So in some ways, seeing mTOR inhibitors come out as a positive effect on lifespan on average, and autophagy inhibitors shorten lifespan sort of validates the approach. And then you've got all the new stuff, which is really interesting, which we're not going to talk about because it's proprietary unless somebody wants to fund the Million Molecule Challenge to make it an open access project.
And what the cool thing about that is, we're starting to use this data set. So again, we're partnering with our partners at Rhizome, and what we're looking at is predicting dose efficacy. So now we've got this data where we've got dosage curves across a significant number of these molecules across these pathways, showing where is the proper target to see an effect, either positive or negative. And now we can start to predict that and start to look at, okay, well, if we're going to do screening, we need to modulate some of these molecules and screen some of them at, say, 10 micromolar, some of them at 100 micromolar, to find, to not lose out on false negatives.
Right.
And that's what this is allowing us to do.
Right. Yep. Okay. So anything we haven't talked about that you want to touch on?
That's a great question. I have a question for you, actually. And I want to know, this is a little bit meant to be not a hot take. I don't think it's a hot take. But so when I think about high throughput screening, the dangers are false negatives and false positives. I've always based my approach that it's so much better to screen out the false positives and only have positives that are worthwhile to pursue. And at some point, you're going to do another screen and look at stuff and do dosage curves and stuff like that. Maybe you'll find out that some of your you've got false negatives. I've always taken that approach. I'm curious, what do you think is worse, false negatives or false positives?
I don't even think I would frame the question that way, because I think they're both limiting in some way. I mean, I think false positives have the potential to lead you and the entire field astray. So in that way, they're worse. Yeah. And just for people who maybe, aren't it's not completely obvious what we're talking about. False positives would be, in the case of a longevity drug screen, you get something that looks like it extends lifespan by, say, 100%, and then you retest it and it doesn't work, right? Whereas a false negative would be, you test something that has the potential to actually increase lifespan by 100%, but in your test, for whatever reason, wrong dose, you screwed the experiment up, bad day, whatever, it came out as 0. So you miss it, right? So both of those are inefficiencies.
I think they're harmful, but
I think it depends on what your goal is. And so this is where, with the Million Molecule Challenge, I'm absolutely willing to tolerate missing some things, because if we measure 1 million things, we're going to find some really, really cool, interesting stuff. Sure, we'll miss some stuff. I also would say the way that what the Ora technology allows for is a pretty happy middle ground, where you can actually do some replicates, and what the replicates will buy you is a lower likelihood of both false positives and false negatives, because you're testing each thing more than once. You can also potentially do doses. But having said that, every replicate you do reduces the number of novel things you can test. So it's always going to be this sort of balancing act.
Exactly.
I think in terms of a given lab or company, you can mitigate the risk of false positives by either retesting, having that built into your pipeline, or validation in, say, a mouse model. Because if it's a false positive in worms and you then take it to validation in a mouse, it's not going to work. So I think you can mitigate it at that level. Where I get more worried about false positives is just, some of the history of the field, and the field being led astray by things, and they're probably not even true false positives in the sense we're talking about it, but by things that one lab reported as really important. And often it's only that one lab that studied it for a while, but it sort of became dogma in the field that this is really important, and people wasted, millions of dollars in careers studying this thing that turned out to actually not be important because the original result wasn't real. Yeah. So that's the big risk, I think, with false positives. A false negative isn't going to do that because you just don't end up studying something that could have been interesting. Right. Fortunately, there's a whole lot of really interesting biology out there. So if you miss one interesting thing, but you're smart and you're doing good science, you're going to find other interesting things.
And I think, we've already shown that. We've done, as we talked about earlier, roughly about 10,000 interventions over the last year with, I'll say this, a team of only about 5 folks. Like, we have churned. And one of the things that I'm really excited about is we've got a couple of dozen really exciting interventions that now we've taken and we're validating across these in vitro cell culture models. We actually are lucky enough, we have a little bit of ex vivo data on human skin samples that are taken actually from patients outside of the context of them on people.
Yeah, and we haven't talked about the cosmetic angle, but I think that's another one that a lot of people are interested in. So you mentioned skin biology, right? Certainly radiation and skin cancer, but these things that affect longevity pathways are going to, in skin, have a cosmetic effect.
And I think that it's really, really exciting to see.
Which you and I both need. So hurry up. At least that's what the comments on YouTube tell me.
What I've always decided is that beards are makeup for men. So as soon as I could grow this, I've never shaved it. My wife has actually never seen me without a beard.
Oh, yeah.
Because this covers up everything. You don't have to worry about it. So that's not to say...
Well, who knows? Maybe I'll grow a beard. I don't know. I've done it a few times in my life, but it's been a while.
I think that, in the cosmetic space, we're seeing cool companies. I know you're an advisor on Hayflick.
Yeah. Right.
Which is Chris L's company out of Drexel looking at rapamycin topically for skin aging.
Right.
Now we're seeing that really cool company focus on this, and I want to highlight them because I really like them.
Yeah, and actually it's worth mentioning. So this is Hayflick Partners, and they are actually going... this could change, obviously, but right now, at least, they're going after wrinkles as an FDA indication. So it turns out that this is actually an indication you can get FDA approval for. Chris has very good data suggesting that rapamycin in skin cream can have a positive effect on skin aging, including wrinkles.
Yeah.
So, I think it's likely that it will work, and it's just a matter of moving this through the FDA approval process to get a new label expansion for rapamycin skin cream.
Yeah. And one of the things I want to highlight, so, we've talked about a little bit difference of context of you are coming at it from the aging field in general, and very much from like the clinical work, the great work you did at OptiSpan, whereas I'm often in the trenches trying to get a drug into a market. And I think one of the things that I love about the aging space is that because it's, relatively new in the biotech and pharma industry, people are more collaborative, and a win for a different company like Chris's going after skin aging, that just paves the way for Ora. And not to overly promote, although I'm going to do this, is that we've got a molecule that works better than Rapa. And so, I want them to succeed in the clinic so that in a couple of years, we can come in with something that is even cooler.
Right.
So,
and if we suddenly start looking 10 years younger in the next round of this video, it might be because we started making our own skin cream with this new molecule.
We talk about having receipts, Matt, and I have my license from when I started in your lab in grad school to now. And I can see the effect that Matt Kaberlein as an aging intervention has on people around him.
Come on.
No, it's it's always been a treat, and it's always been, I'm always grateful to get a chance to chat with you and work with you.
Yeah, I think we can at least say being a graduate student in my lab does not age people as much as being president of the United States does.
That is true. Luckily, neither of us are running for office.
Yes, that's for sure. Awesome. Okay, so this has been fun. Where can people find you, find out more? We mentioned the Ora website, but...
So Ora website, LinkedIn, Twitter, X, whatever social media that you want to reach out on. I love chatting. I love talking about this. If you want to talk about what we're doing at Ora, I always highlight if you're an educator, want to talk about getting your students access to cool data, please reach out. If you're an investor and want to help fund the next generation of longevity therapeutics, this is kind of the inflection point that Ora's at, where we've got our cool pre-lit clinical data that's translating from our in vivo to in vitro. And we're hoping to get stuff into phase 1 in about a year, maybe a little bit longer. A year is pretty optimistic, maybe about 18 months. this is kind of the coolest time to get in and join our team.
Awesome. All right. Well, thanks, Ben. As always, thank you all for watching the OptiSpan podcast. If you've got any questions or comments, leave them below. And if you're not yet a subscriber, are you doing? Come on.